Howdy y'all. Thank you everyone for coming here and seeing all of our presentations. So for my research project, I was the Living Docs intern, and what I was looking into was alternatives to plastic in our Living Docs program. To start, I want everyone to just close their eyes for a second and imagine what a dock looks like to them. And when you open your eyes, to the left is something that you'll commonly see. All you really imagine when you think of a dock is a walkway to your boat, and that's about it. And when you look at these pylons, all that you really see are some barnacles and algae, and that's about it. However, the living docks are so much more. The calcium carbonate substrate allows for higher recruitment levels of a more diverse spectrum of benthic filter feeders, and this has so many beneficial implications. Just to dive into a little bit of the history of our oyster program, before we created the Living Dock mats, we did bag oyster shells and stuff of that nature. However, that program has started to fall on the back end because the one drawback of creating those oyster reefs is they go on the ocean floor. And as runoff comes from upstream, muck and sediment begin to build up on those oyster reefs and slowly start to suffocate these entire areas, which isn't really beneficial when you're trying to look for a long-term solution. So when you look at this mat, right now it kind of just looks like a jumbled bunch of pixels. However, there are so many different things on here. Right here, there's some raspberry tunicates. Over there, there's some solitary tunicates. We got some fire sponge out in that area, and then some barnacles there and really all over. Additionally, we have encrusting bryozoan on the mat itself, which is really interesting to see because it shows that the entire mat is being utilized when on the pylon. There's some colonial tunicates, we got tube worms, sponges, and last but not least, oysters as well. So we can see that these are amazing mats and they're capable of doing so much for us. But the main drawback with these is that they create so much plastic waste. In fact, just creating 3 of those mats created this much plastic waste. So when we're considering a long-term, large-scale solution for this program, we want to try and think about alternatives to plastic. The main drawback of plastic is that when they're put in ocean environments, erosion begins to start. And this has many impacts, not only for us, not only for the animals, but also for us. A study that I was reading shared that 10% of animals in the study were found to have plastic in their digestive tracts. And that goes up the food train and ends up in us as well. And when these plastics are on a microscopic scale, they've been found to go through cell membranes, and that can have catastrophic health impacts for us. And last but not least, when we're doing conservational research, we don't really want to contribute to the pollutional problems that we're trying to solve. So we kind of have a moral obligation to stray away from plastic if possible. So the real question is, what are the alternatives to plastic in the Living Docs program? The 4 that we came up with for this study are galvanized steel, jute, which is a plant-based fiber, Bessie, which is a potato starch zip tie, and coconut coir, which pretty much speaks for itself. The 2 questions that I came up with for data collection for this study is, at first, what organisms will settle on different materials, as well as how fast will each material degrade in different environments. To answer the first question, I pulled from a study that I had read, and it ascertained that coconut coir was a great replacement for plastic because it increased recruitment rates exponentially. And then to answer the second question, I personally believe that galvanized steel would be the worst by far, just because of corrosion in saltwater areas. And I believe that it would rust at a really fast rate, especially because they were very thin wires. So, for this study, it was in the Indian River Lagoon at the bottommost part in Martin County, and right next to FOS. So for this project, we picked 2 different sites, a high and a low salinity site, and we chose them based off of their proximity to Earlon stations, which you can see in the blue mark. These Earlon stations provide consistent and reliable data, water quality data that we're capable of relying on, so that we can look at them and base all of our other data that we're collecting off of. So, for each of these sites, we put 4 mats of each material out, adding up to a total of 40 mats, which took a long time to make by hand, and I could not have done it without the research team. Just to create one of these mats, first what you have to do is grab a 2x1 polyethylene mesh. You then have to drill 30 shells for each mat, and to dive into a little bit of the math for this entire project, we had to drill an entire 1,200 shells in total and fasten each one of those to the mesh. Then, after that, we had to weigh 6 fastening materials for each for data collection throughout the study. At that point, I would then randomly select 6 positions on the mat for these weighed fasteners to go on. At that point, I would then begin the assembly process, where I would create 3 rows of 10. After that was complete, I would then attach 6 colored zip ties onto the weighed fasteners that I had weighed at the start. And then I would then cut off the excess from each of these mats. So I could then have the starting weight before deployment, so I could base all of my degradation data off of this number. The timeline for the study is as shown. It's an exponential study, so we started out collecting data 2 weeks after the experiment was deployed, and then we would go 4 weeks after that, then 8, 16, 32, and 64. However, for my study, I was only able to collect the first 2 data points because of time constraints. However, I was still hopeful that I would get some trend from this and I would be able to determine some conclusions, just because 3 data points is still statistically significant. For the data collection process, it was pretty simple. All we had to go and do was go to both sites, take a single oyster off of each and collect the fastener. And then at that point, we would go back to the lab, analyze each of the fasteners for presence or absence of different filter feeders. At that point, I would then begin cleaning each of the fasteners to get all of the organic material off, or as much as I could, so I could determine the degradation rate, so it wouldn't be impacted by the organics that had been fouled onto the materials. And then I would toss them in the oven so I could get the dry weight to compare to the starting weight before deployment. And at that point, I would then analyze the data that I had gotten to find the trends. So, when asking the first question, what organisms will settle on these different materials, we performed many statistical tests, and we found that although the data was statistically significant in multiple ways, I wasn't able to create a visual format that was really efficient in showing anything significant. So I created a data, just a little table to show you guys what was on those, the top 3 at each site for each material. And I was going to share a couple interesting things that I found. First off, biofilm is everywhere on everything, kind of just as the first thing to go. And then also, on both of the fibrous materials, coconut coir and jute, mussels were really loving them for some reason, and they would just attach to them and out of like crazy proportions. So it was really interesting to see that. Going on to my second question, asking how fast each of these materials will degrade in different environments. We performed a Shire-Ray hair test and determined that this data was statistically significant. First off, looking at the plastic, our control, it was actually really interesting because there was an increase in weight among all of them, which was kind of odd to see at first. However, when you think about all of the organics on these materials, I'm only human. I can only get so much off. So the little nooks and crannies, I couldn't get every single last bit of it off. So they actually increased in weight after the experiment began. Next, looking at our worst performer, jute. We had 100% loss at the 6-week point in our study, which was really interesting to see. The only shell on any of them was the one that we had replaced at the 2-week period. So it was really cool to see that something just absolutely blew everything off except for the very few that we replaced at the first data point collection. And then going on to coconut coir, we saw that there was a higher decrease in material weight at the low salinity site. which is contingent with our BESSY as well, which was the potato starch zip tie, if you guys forgot about that. And it was interesting to see that these both were very similar at both the high salinity site and the low salinity site. And last but not least, the stainless steel was our best competitor. And it showed that there was 0% loss on any of these, which was really interesting to see. So we'll see how it goes later on in the study as they begin to sit in the water for longer, but I'm hopeful that they won't really corrode much at all because they haven't showed any signs of falling off. We also looked at and saw that both BESSY and coconut core were statistically similar, as well as plastic and stainless steel. However, jute is literally off the charts. So, going back to my questions that I started with, looking at the first one, asking what organisms will settle on the different materials, I determined that I couldn't really accept or reject this hypothesis, just because I hadn't collected enough data at this point to really create any conclusive results. So then, looking at our second question, how fast will each of these materials degrade in their different environments? Stainless steel was clearly not the worst. It was in fact the best. Jute was gone. So I had to reject this hypothesis, which was a very happy rejection at that. So what is the main takeaway of what I've been doing this entire summer? Stainless steel is the best replacement for plastic. However, there are still some drawbacks from using this instead of a much more easy material to work with, like plastic zip ties. For each of these, I had to use this little doohickey to fasten on each of them, and although it might look really painless right there, each one of those took like 5 or 6 pulls to really get it all the way twisted around. And also, I had to use a glove because you needed to put a lot of energy into holding that down. So the time, energy, and cost that it took to create just one of these mats was double, if not more, from creating something like a plastic mat. So it is still worth noting that plastic is still an incredibly cheap and effective fastener for this program. However, environmentally catastrophic it may turn out to be. A couple shout outs. First off, Morgan, thank you so much for helping me throughout this entire study and showing me the scientific process. It has been invaluable. Also, the entire research team, thank you guys so much for helping me make the mats, deploy the mats, and collect data throughout the entire process. You guys have been so helpful. The entire FOS staff, thank you guys for hanging out with me and, I don't know, being great coworkers. And then the FOS community volunteers, you guys truly made this project way less painful because although there were 1,200 shells that needed to be fastened, I didn't need to drill every single one of those. So that was really nice and would have made this way more painful. And last but not least, I forgot to talk about this at the beginning, but this study that I was performing is also part of a much larger study that will be followed up with Florida Tech. Dr. Kelly Hunsucker is leading that this project, and I'm just a small part of this much larger project. So I'm really hopeful to see what will come of it. Does anyone have any questions?
Fantastic presentation. Just a question, I want to hear your thoughts on it, not that there's a scientifically correct answer or not, but
yeah.
How would you weigh the benefits of stainless steel in terms of durability versus the potential environmental impacts of putting something out in the water that is a forever product? Stainless steel is going to be around for a really long time.
Yeah.
Good if you want it to last, but bad if you don't want it to last. Where do you think your head is at on that?
So, although it is more energy intensive to put them on there, I think the environmental impact and what you can just wave off your conscience as a scientific conservational person, when doing that, although it might take more energy, and if we want it to be a large-scale volunteer event, we'll need to get a bunch of these different tools to do that. However, I think the cost of putting plastic in our oceans and knowing what it could do, whether that's in 5 years or 100 years, it's going to be there for forever after that, and it's impossible to clean up these microplastics when they're at such minuscule amounts, and they will end up in our bodies. So it's kind of just, you're weighing an economic cost versus like a moral obligation to be a better person, and like it's not that much energy in the short term, like in the short run, to do something much more beneficial for the environment in the long run, you know? What's that?
So they're not going to be doing the oyster bagging anymore? That's not working well?
It's currently on hold. In the back of the property right now, if you go on the trail, you can see like 5,000 bags on the bed. So we want to continue it, but I think at the current time, we just don't need to make more, especially when the drawbacks of putting more plastic in our ocean and the oyster reefs in general, they're effective, but not as effective as these living docks have shown to be. So, yeah.
How has it been going where they cast it to the piers?
They've been going like really well. There's actually one back there, and all of the ones that we've looked at that have been out there for 6 months, they're crazy. There's so much stuff on them. Like the picture I showed you guys at the start, that had so many different things on it. There were crabs too. There were so, like, I didn't even like scratch the surface of what all is actually on that. I didn't even have enough space. So if they're really effective, and we attach them with huge zip ties on the back too. So once they're fastened on there, the great part about the living docks as well is that these different benthic filter feeders attach to the dock as well. So they cement themselves and help with the fastening process over time. It's just something that you need an initial fastener, and also when there are seasonal changes, different species might die off at a faster rate, and like there are seasonal changes in what is recruited onto these mats. So we need something reliable to keep the main shell on, but it's awesome to see that when these things, if you try picking it up, it's like 50 pounds or something crazy. Probably not 50 pounds, but it's really heavy. So there's a lot of stuff on them. Basically, it's really great. Long story. What's up?
Do you think using a mixed material kind of method could increase biodiversity on that?
A what material?
Mixed material, like some stainless steel, some jute, some messy.
I'm curious to see how the rest of this project goes, to see if there, we determined when we ran stats that there was a statistical significance between different materials at the different sites. However, I couldn't like visually show it in any good way, just because there's like, it's just hard to do that. So I think once we get more data, we can definitely determine if it would be beneficial to add different things on there, so we can recruit mussels with coconut core, and also have like the strength of stainless steel and stuff like that on some of the other parts. So it'll just come with time, really. What's up?
Kind of a question for all three of you. You listen to the news says that the waters in Florida are boiling.
Yeah.
Do you think that your experiment or any of the other two experiments would have different outcomes? I don't know. Is the water temperature higher here now than?
I mean, I can't speak on the history of this area. However, this summer, sometimes when we're in the water, it's really hot. Like, I'm getting heat strokes, and I'm like getting woozy and stuff.
Don't say that out loud.
Well, no. I also hydrate. We also hydrate a lot. I'm not like, well, all right. Oh, jeez. But I think Paige could probably speak to that best, because her, like, seagrass is a very fragile species, whereas like benthic filter feeders just range such a large amount where something will attach to these mats no matter what. It just depends on what it will be. So seagrass is a much more finicky organism.
Yeah, I thank you for your presentation. I was just wondering, since you're not going to be putting in oysters because of the buildup of the sediment, what about clams now? Are you going to, because I know you started to do some work here, I read you were doing some work here in the facility. So would that be an alternative now that you would use for this type of experiment?
I can do that for you. So just to clarify too, we are all for oysters and putting oysters out. This past year, DEP has really cracked down on us, and so that has been everyone. Yeah, everyone. Not just us, but that has been the slower end of the process. It has taken us 6 to 12 months to get a permit this past year for an oyster Yes, and so that is why we slowed down.
But the intent is to maybe look into that aspect, or is you're just waiting for permits, or is that all?
Another thing down here, seagrass has been doing really well, so you can't plant an oyster reef if seagrass is present.
Okay.
And so, yeah, that's a good thing for seagrass, but bad thing for planting oyster reefs. But we are doing other work, part of Paige's, off of hers, with plants and seagrass.
But the
yes.
question, I just want to make sure I understood, was that since the oysters seem to have other negative benefits, let's say right now, would clams be the alternative way to?
I think it depends on where you put the oyster reefs. So obviously there's more buck accumulation in canals and freshwater runoff from that, right? So it really is site dependent. So depending on the area, your oysters can be very successful. We have a very successful oyster reef in the mid-estuary right now. Yes.
And going along those oysters and clams, they are in different parts of the water column, right? So clams are in the sediment, oysters are above, and so they perform the same ecosystem service of water filtration, but they do it in different places. And so ideally, we would do it all together. Like we would have clams and oysters, but because of permitting, because of like restrictions in habitat, some places we can go and we can put clams in and it does well. Other places it can only be oysters. So it just really depends.
I was just envisioning that with clams, you just throw them in water.
Yeah, yeah.
Right? With here, you're building a reef. So to get around the sediment issue, that might offer you an alternative, and I was just wondering if you were.
So one of the downfalls of clam restoration is you throw them in the water, but you have to protect them from large critters. And the way you protect them are with huge plastic mesh nets. So, like, what do we do with that?
Just pros and cons to everything, I guess. So, anything else? Yeah.
Is there any push by Forest Graphic with municipalities to be able to put the living dots or the mats on like bridge pylons or any other public docks that are around to be able to utilize those? Or is it mostly private docks?
Yeah, so we work closely with the community, but we also did one at Clifton Prairie Park recently, and then we had our summer camp help us deploy them to that, so that's really nice to be overall involved.
So at Lawrence County, we did one up in Indian River
Oh, that's
County.
actually good.
We've done some at some larger docks in St. Lucie County. So this is just a very small subset, but Morgan has her hands full. She has docks all over the place. There's many more to come.
Not to mention the ones up in Clark County, Florida Tech too, so there's ones up there as well.
In total, you guys have deployed like 40 or something, right? Like 40 different docks.
Oh, 30 for our pilot study, yes, and then including the ones from Florida.
Okay, that's good.
Sorry to cut back off. Feel free to talk to me after. Thank you all for coming out today. We're very proud of our interns. They were only with us for 12 weeks, and you've seen the studies that they were able to do in just that amount of time. So can we get another round of applause?